Biology is magical. And the moment I say pluripotent stem cells, the whole world of magic begins. Hey everybody, welcome to the webinar. My name is Shekhar Suman. I'm the CEO Managing Director of Biotechnica. And today we are going to talk about career in stem cells. Now you will ask, why exactly suddenly we are talking about this? Because this is the groundbreaking revolutionary technology of the future. Thanks to all the research happening around the world, it is happening now. And with the advent of AI, today we are reaching a stage where potentially in the future we will be able to reprogram any cell into a stem cells. Well, don't believe me when I say that. But our expert of today is going to make you realize how stem cells, which the research we started a few decades ago, is going to explode in the future. And in fact, even today. And the best part, we have the best university, the best college in Asia for regenerative medicine and stem cells right here in Bengaluru, India. And today I have an expert from the same particular college, Manipal Institute of Regenerative Medicine. Today we have Dr. Sveta Chakrabarti. If you could please bring her up to the stage. Hi, Shweta. Good evening. How are you?
Good evening. I'm doing well, Shekhar.
Yeah, thank you so much for joining. It's a huge pleasure for all of us for hosting you today. So, like I was saying, Dr. Shweta, I'm sure you have a big presentation to share with all our audience. But before we get started, what's your outlook about the stem cell technology and how do you think it is going to pan out in the future?
I think the stem cell technology is moving really fast. Of course, as you mentioned about AI, maybe it's not
Right,
moving as fast as AI because we are working with cells and
right.
dealing with any living system comes with its challenges. And of course, it's a system you can't really always control as you want. But
Right,
definitely, as you said in your introduction, with the advent of these induced pluripotent stem cells, we have
right,
now the possibility of making any cell type in a dish. And that's
right,
opened up the avenues of both research and disease
right.
outlook has become very much exciting for us researchers. Yes.
All right. So now that the stage is set, we have understood the importance and the growth potential of stem cell technology. Let's talk about the topic of today, stem cell careers, and how it is going to help our audience. So the stage is over to you, Dr. Shweta. Please go ahead, share your screen, and let's get started with the webinar. And at the end, we have two gifts for our audience, guys. Please don't leave the webinar in between. If you are attending this webinar, and our team will be sharing a form at the end, in fact in between also they might share, if you click and fill that form, you're going to get a free certification course on stem cell technology, free of cost from Biotechnica. It's worth 1,000 rupees, but it is going to be free of cost for all of you if you register today. So attend this webinar till the end and fill that form. So Dr. Shweta, over to you. Please go ahead and educate our audience on the careers and potential of stem cell technology. Thank you.
Thank you. So first, I would just like to check whether everybody is able to see my slides. So if
Yes,
that's
we can.
yes,
We
okay.
can.
So then I'll start with my presentation. Thanks for the introduction. As you have heard that I am a faculty at the Manipal Institute of Regenerative Medicine. We are at the Manipal Academy of Higher Education in Bangalore. And I'm really happy to talk to you about the topic of stem cells and regenerative medicine and what we are doing here at our institute in terms of the teaching programs, but also in terms of research. But before I step into that, I thought I would take some time to tell everybody about stem cells. And so I'll actually begin my talk with the early division in the one cell zygote. So as you can see from the image on the top, which is taken from a nematode, which is the C. elegans nematode, that is the C. elegans embryo. This C. elegans embryo has been labeled with two different proteins, one in green, which is the microtubules, and these are basically helping to segregate the chromosomes in these zygotes, and the chromosomes are labeled in red. And as you can see, as you progress in mitosis, the chromosomes are separated, and we are easily able to observe them under a microscope because this organism is transparent and we can see this very easily. Similarly, on the bottom, you have cell division during what is in a somatic cell. So on the top is a zygote, and at the bottom is a somatic cell coming from humans. So similar microtubules in magenta and the chromosomes in green are being separated first. They're being aligned in metaphase, separated during anaphase, and then finally during telophase, and cytokinesis that's happening in the last phase that you can see. So this is cell cycle and cell cycle progression that happens in all dividing cells, including stem cells. But what's so special about stem cell? What makes a stem cell and not a somatic cell that I've shown you in the bottom? Well, to become a zygote first, what do you need? You need a haploid male gamete. You need a haploid cell that is not having both the copies, but one copy of the genetic material from the males. And similarly from the females, they need to come together to form the zygote, which is the fertilized egg. But this zygote is a very specialized sort of stem cell. It has the ability to actually divide and differentiate to give the entire body plan of an organism. So here on the right, on the top, you can see how at 28 days, this zygote has divided to form sort of a body plan that you can make out. But by 23 weeks, that is around 50 days, you start to see all the different tissues and organs forming. And that's in the case of the humans. But of course, we cannot study this development and embryonic development in humans. And so we use fruit flies as a model system to do this. And this video at the bottom will show you how we can actually look in living tissues at development of the zygote to give rise to the entire organism. So this is about 4 hours post egg laying, and you can see already that the germ band extension is happening. You can see all the segments are forming. So gastrulation is proceeding. You can start to see the different segments forming on the bottom panel, and now the cells are moving and they will be closing together. These are all the cells that are moving from both the sides. So in the dorsal view and the top view, they're now fusing together to be able to give rise to the proper germ band extension. You have all the different thoracic regions and the segments being formed. And soon after this, you actually see that the embryo will start moving very vigorously, and that's the hatching. And from here, the larvae will emerge from this entire embryo. So you saw on how the syncytium can now change and give rise to the entire larvae. So the larvae will be hatching from this embryo, and we will no longer be able to see it into view. But this tells you that these specialized stem cells give rise to so many different kind of movements. And this is really incredible that we can study this in the lab and understand how this process takes place. Now, this is the same C. elegans embryo that I showed you stills of. This is a live movie. You can see the mother and the daughter nucleus meeting together. They're fusing together. Now in the middle, you can see now the chromosomes are being segregated, and now two cells are being formed. This division is very peculiar because it's an asymmetric cell division. The left anterior cell is larger than the right posterior cell. And as you can see, each cell is now dividing to give different sort of cell shapes, and the contents will be different. And as this movie will play out, you will see that you will actually go from 1 cell to 2, 2 cells to 4, 4 to 8. And later on, this movie is cutting off at 46 minutes, but this will give rise to the entire organism, which is a worm which is found in soil. This is a nematode called Caenorhabditis elegans. But because of the transparent nature of this organism, scientists have been able to understand embryonic division and development in this organism with ease under the microscope. So this is a simple light microscope, and by just mounting the embryos, you're able to understand how important asymmetric cell division is in forming the cell fate decisions. So that's in the first cell division in C. elegans. I want to show you that though in the first movie you saw that the sizes were different in the cell division, but that's not all. actually proteins segregate very specifically in these two different cells. So you start off with a mixture of proteins, but now you can see on the left cell, you have the protein which is labeled with a cherry molecule in par 6 that's segregating on the left, and par 2 which is segregating on the right. And in fact, if this segregation doesn't happen into the lipid layers, then the embryo cannot give rise to the entire organism. So the segregation into the different daughter cells is essential to actually the development of the organism. So it's not just a simple act of forming two cells, but the contents of the cells have to be segregated very specifically to give rise to the entire organism. So I've been talking to you about, basic organisms like the worm or flies, but how about other stem cells? These are the embryos. How about other stem cells in other locations in the body? So the role of stem cells is important not just in the context of development, but it's also important in tissue repair, in tissue renewal, and in regeneration. And what's so special about stem cells is that the stem cell can divide either symmetrically to give rise to another daughter stem cell, or asymmetrically to give rise to a differentiated cell. And the first cell that you see here in purple is a kind of totipotent cell, which can give rise to all kind of different cell populations. But then later you can have multipotent cells, which are in green or in blue, which can give brown, which can give rise to very specific kind of cell population, but also have the capacity to divide, and they will differentiate. So that's something that's very special, which is the property of stem cells, which no other cells in our body can do, and only this particular type of cell can do. But as I said, what about the stem cells which are found in other tissue? Well, here on the left, you have a Drosophila neuroblast. So this is a cell that can give rise to neurons. And this is marked with two proteins, a membrane protein PON, and the DNA is in red. And as you can see, as this Drosophila neuroblast is dividing, again, the DNA is condensing in the middle and a metaphase. And then as it's about to divide, you have this segregation that happens to a stem cell, which is marked by PON, and the differentiated cell, which loses this PON expression. So retaining the PON will make it a stem cell, and losing it becomes a differentiated cell. And the way we know this is important is mutants that are affected in the same pathway. So there is this mutant called BRAT. In these mutants, the stem cells never stop dividing. They don't divide asymmetrically, they divide symmetrically, and you get this phenotype where the brains become very enlarged and look like tumors. So these organisms don't develop properly and actually die during development. And so it's not just important in terms of segregation and differentiation. In the loss of these factors, you can even lead to tumors in an organism like the fruit fly. And here you can see the brain tumors in these particular mutants of this protein called BRAT, which is important in asymmetric cell division. So what happens in these mutants if you try and transplant tumorous tissue? As you can see in this experiment, GFP label cells have been transplanted either to wild type flies or into mutants of the asymmetric cell division pathway, whether it be this gene called RAPS, Mira, which is Miranda, or NUM. And what you see is that in a normal wild type fly, the tumor doesn't really grow. It stays within the same location. But in these mutant backgrounds, you can start to see these GFP labeled mutant clones growing in the entire abdomen, and they can take over the entire fly and lead to a lethality. So a loss of these asymmetric cell division regulators can actually cause tumorigenesis and the progression of tumorigenesis in the fruit fly. And of course, we know this also in higher organisms, and I'll be talking to you this in the later part of my lecture. But going back to what are stem cells? So as I told you, stem cells have the capacity to divide, to give rise asymmetrically to a self-renewal, that is, give back a stem cell, but also differentiate to give rise to a differentiated cell, which could be any somatic cell. So a stem cell is very special, and it's called multipotent. But you can also have transit amplifying cells, which are called progenitor cells. These are multipotent in the way that they can give rise to very different kind of terminally differentiated cells. As you can see in the colors, you can have from these orange to yellow to green to blue different cells. And these can be arise because of these multipotent progenitor cells, and you can have unipotent, which are only able to form one particular type of terminally differentiated cell. But the stem cell is special because it can give rise to all of these different colors of stem cell, as you can see here on the right. So that's the special thing about stem cells. Now, taking an example of adult stem cell, I would like to kind of focus into one tissue which my lab works on, that is the gut. Now, in the gut is a very specialized structure. As you can see from the scheme here of the small intestine on the top, you have these villus-like structures, which are these absorptive cells, which make the gut able to absorb a whole lot of nutrition. But what's special is this crypt, which is on the bottom that you can see, are these stem cells are actually at the bottom of the crypt labeled by red, and this can give rise to the entire villus. So what's special about them is that they can give rise to these transit amplifying cells, and they can differentiate into all the cell types. So what are the different cell types that you find in the gut? In the gut, you have the absorptive cell, which are called the enterocytes, which actually can absorb the nutrient. You have the goblet cells, which can actually help in digestion. You have the Paneth cells, which help in maintaining the niche, which is very sterile, which is important in producing antimicrobials that protect the function of the crypt so that no microbes can get inside the crypt to keep the stem cells away from any microbes. And you have the signaling cells called the enteroendocrine cells. These are all the specialized cell types. These are EM images, drawings from EMs. And you can see that the morphology is very, very different. But a single stem cell is able to give rise to this whole different battery of cells that you can see in the gut, which have very different functions. So how was this discovered? Well, there were very nice experiments that was done in the lab of Hans Clevers, where they were able to label specifically these stem cells at the bottom of the crypt using specific transgenes, and these can be marked with a gene called LGR5. LGR5 can mark these specific stem cells, and by labeling them, they were able to see in the mouse gut how one cell can move up as a conveyor belt all the way to the top. And these transit amplifying cells can then keep dividing and moving up, migrating and differentiating. And finally, at the top, you have the cell death that happens. But as you can see on this scheme on the right, LGR5 stem cells can give rise to either stem cells or Paneth cells, but also these transit amplifying cells that are able to give rise to all the different populations that I talked about just before, the goblet, the enteroendocrine, the absorptive enterocytes. And all of this happens under a very important pathway, which is the Notch pathway. In the absence of Notch, LGR5 cells are not able to differentiate, and they stay as stem cells. So it's actually the Notch pathway activation that is important for making this differentiated cells.
The lining of the small intestine, like the lining of most of the
So this is just a video that's showing the stem cells. I hope you're able to follow this.
The epithelial cells, usually called enterocytes, help absorb nutrients from the lumen of the gut or absorb water from the intestinal contents. In the small intestine, the surface of the lining of the gut is increased enormously by thousands of villi that protrude into the lumen. Here we see a single villus and its internal architecture. The surface of the villus is covered by a single layer of enterocytes, which extend down into the crypt below. Stem cells at the bottom of the crypt, located between Paneth cells, divide and make copies of themselves, and also make transit amplifying cells. The transit amplifying cells proliferate rapidly and move up the walls of the crypt. As the cells migrate upwards, they begin to differentiate into goblet cells and enterocytes. While the cells are moving up the sides of the villus, they carry out the essential functions of the small intestine, notably absorption of nutrients. When the differentiated cells reach the tip of the villus, they undergo apoptosis and are shed into the lumen of the small intestine. The entire process of outmigration and cell death is completed in just 3 to 4 days. The process of outmigration and rapid cell replacement is a defense mechanism against the development of colon cancer, since almost all epithelial cells, including those that have accidentally sustained mutations, are shed within days of their formation. Therefore, the only mutations that can lead to the development of a cancer are those that are retained in the crypt. This dictates that such mutations must block the outmigration of mutant cells from the crypt. The outmigration of transit amplifying cells from the bottom of the crypt depends on a protein called adenomatous polyposis coli, or simply APC. In the absence of functional APC, this continuous outmigration is blocked, leading to the accumulation of transit amplifying cells in the crypt. In this animation of an experiment, APC loss is achieved through an induced gene inactivation, which appears to mimic the mutation that initiates most gastrointestinal tumors. APC is usually required to inactivate the intracellular protein called beta-catenin. The inactivation of beta-catenin permits the differentiation of the transit amplifying cells and their continued outmigration from the base of the crypt. In the absence of APC function, beta-catenin accumulates within the transit amplifying cells, which blocks both their outmigration and their differentiation. The accumulated transit amplifying cells do not themselves form a carcinoma. However, they and their descendants can now accumulate additional mutations that will drive such cells progressively to become full-fledged carcinoma cells.
Okay, so that video very nicely showed how these mutations that can occur in stem cells in this protein called APC can lead to adenomas. So you can see here, this is a picture from a normal colon on the left, and now you can see on the right where APC gene is mutated. As you can see now, these transit amplifying cells, so that was just like a cartoon representation, but this is an actual tissue coming from a patient. As you can see, the normal colon looks the way it is organized on the left, and on the right, when the transit amplifying cells are not moving up the villus and are staying there and start accumulating mutations, this is what can happen to the structure of the gut. So this is how we get to know that proper control of cell cycle and the Wnt pathway is very important in the differentiation of these transit amplifying cells. And these were first clues that told us that not only the Notch pathway, but also the Wnt pathway is very critical in the behavior of these transit amplifying cells in the gut. But coming back to the stem cell technology and what we can do with developmental biology and how we can move it forward to kind of trying to recapitulate these experiments in the lab. Of course, working with mice does reveal a lot, but making mutations in the crypts LGR5 can be quite complicated, time intensive, and can require a lot of resources. What researchers have been able to do is label the LGR5 cells specifically in mice, isolate them, and actually grow them in the lab to form these kind of in vitro mini guts that you can grow in dishes. So you can recapitulate the entire gut morphology in a dish by just taking LGR5 expressing cells, putting some differentiation factors over days. In about 8 days, you can start to form lumens and these sort of mini guts. So this has been really a big development that has happened in research that has been able for us to recapitulate what happens in the organism in a dish. And that has led to a lot of gene editing technologies being used here and a lot of medicine that's coming, and I'll be talking to you about in my next few slides. So what was the discovery done? How was it done? This was done in the lab of Hans Clevers by Sato when he was a postdoc in Clever's lab. Now he has his own lab in Japan. So what he did was he identified these LGR5 cells. So they found this LGR5 gene. They actually marked it with GFP. So they could isolate crypts from mice, and then they dissociated the crypts and isolated individual GFP labeled cells, as you can see in panel C. Then they grew them in a specialized way, which was in this matrigel, which kind of can make the environment conducive for these LGR5 cells to form these kind of bodies. And then with differentiation factors, as you can see in panels E, F, and G, he then differentiated them to form these guts or mini guts in vitro. So this was the first demonstration. Later on, people have recapitulated this for other cell populations as well, and even for brains. So people have even been able to make mini brains. And if you have LGR5 or the stem cells from the human gut, if you're able to isolate biopsies and get the stem cells from the crypts from a human, you will be able to recapitulate the gut morphology from a human in a dish. So this basically gives us a handle of working with a tissue without having to work with an organism. We can make the tissue in vitro and test it with different drugs and do gene editing on it without having to go back to the organism. So this gives researchers a really big handle to be able to understand the biology, but also to figure out mechanisms to heal diseases or to work out strategies to, help in disease progression, to not when we saw in APC, maybe some molecules that can prevent the beta-catenin function and help the transit amplifying cell from differentiating. That could be one example that could come from these sort of experiments. Now, some of the very important contributions that have come in the field of stem cells, and then I will be talking to you about induced pluripotent stem cells, have been from the labs of Thomas King and John Gurdon. So these were actually experiments done in the 50s. So these were a long time ago, where they showed that you could make stem cells by doing nuclear transplant experiments. So you could take eggs and you could transplant the nuclei from blastula cells into these frog eggs and then reprogram them. And this was done even in Xenopus levis by Gordon Lab, where he took somatic cell nucleus. So he isolated the nucleus from epithelial cells and put them into the embryos, and then he was able to, make a full organism of it. And for this, John Gurdon also received the Nobel Prizes. But after that, in the 80s and in the 90s, there have been a lot of development in embryonic stem cells from mouse. So a lot of research have moved from looking at frogs, then to mice, and then finally to human embryonic stem cells. So in the early 80s, Martin Evans in the University of Cambridge was able to isolate and grow mouse embryonic stem cells. He won the Nobel Prize for this in 2007. He developed gene targeting technologies and to create animal models to study human diseases. Then Thompson and Gerhardt isolated human embryonic stem cells and were able to grow that in the lab. So this was a big landmark discovery that was done in the field of stem cells. And a really big discovery is done by Shinya Yamanaka in 2006, where he was able to reprogram any somatic cell into a pluripotent cell. So that was, again, a really big discovery. And his discovery has then led to the use of iPSCs, but also the human embryonic stem cells in the treatment of spinal injury in 2010, in 2012, to help in the treatment of blindness. And now even in trials, induced pluripotent stem cells have reached to that state that I think in the 2020s, people have been trying to use that again in the treatment of blindness and other specific diseases, such as multiple sclerosis and other diseases like that. But as I said, there was this experiment done by John Gurdon back in 1950, which only won him the Nobel Prize in 2012. So this is just a point I want to give to the young audience out there, that it can take up to 50 years for your contributions to be recognized, and the application when Shinya Yamanaka could show that you can actually reprogram human cells and make them useful for technology. But this landmark discovery was done as early as the 1950s. So in science, perseverance is very important. And that's something, at the young generation, I would like to give them as a message that in science, perseverance is important. And Sir John Gordon contributions were only recognized by 2012, but in the scientific community, we were studying looking at his contributions in all these years. So this is his beautiful experiment that he did in the 60s. He actually took skin cells in a culture dish, and with pipetting, and because of his really good technique, he was able to isolate just the nucleus out of this skin cells. What they took was an unfertilized egg because the eggs are laid outside in Xenopus, it's easy to manipulate them. They did UV light to destroy the nucleus of these eggs, and then transplanted with this micropipette the nucleus from the skin cell, and this then led to the normal embryogenesis that I showed you in my first few slides and give rise to a tadpole. So as you can see, that is a very simple experiment, but a very elegant experiment, shows that this egg has all the information required to reprogram the nucleus from even a skin cell to go back to making an embryo and a tadpole. So I just wanted to, show you this kind of experiment that Gordon did back in the 60s. And then people in the 90s were able to do something similar. They were able to take out the early blastocyst. So in the early embryo, which is called the blastocyst in mice, they were able to extract the cells from the blastocyst, then culture them in vitro as these embryonic stem cells. And then after culturing, they were able to put it back into another recipient blastocyst and make these kind of chimeric mice. So you take cells which are sort of labeled in RFP or GFP, put them in an unmarked mice, and then you would see in the adult foster mother, you would see the tissue coming from both the mother as well as these injected embryonic stem cells. So this was also really a great technology that was done in the 90s and in the early 2000s. So what about induced pluripotency, which you heard about the introduction? Why is this making a huge noise in our normal lives as well? Well, it's been because of these Yamanaka's magic factors. And I'd like to show you a little bit about his experiments. What he did was he took these normal fibroblasts, you can see these triangular cells, and he selected, based on literature, some 24 transcription factors. He cloned these genes and expressed them in these fibroblasts, and then what he saw was expressing these 24 led to formation of the same morphology that you see in embryonic stem cells. And then he could differentiate them back into the fibroblast again. But how did he get those 4 magic factors for which he got the Nobel Prize? Well, it was a really systematic screen that was done with all the 24 factors, and where he did a group of 1, 2, 3, 4, and combinations of 2s and 3s. And finally, what he discovered was a combination of 4 transcription factor, namely c-MYC, KLF4, SOX2, and OCT4 were sufficient to cause the formation of pluripotency as all 24 factors did. So just these 4 transcription factors could recapitulate what all the 24 factors could do. And he discovered that actually in this transcriptional network of KIF4, SOX2, OCT4, which forms this kind of a positive feedback loop, as well as MYC, helps in keeping the pluripotency. And you can reprogram any somatic cell by expressing these and keep them pluripotent in the lab. So I just wanted to tell the audience that how discoveries are made and how much perseverance again is required for you to systematically go through all the different transcription factor to get these magic factors that he got. But iPSCs or embryonic stem cells have this propensity to make any sort of cell. So what you can do is you can either grow them in a monolayer, as shown here on the left. But if you now put them in a drop, they form this embryoid body. And then based on different goodies, you can give them retinoic acid, insulin, you can make them into adipocyte. If you give them just retinoic acid, they form neurons. If you give them macrophage colony stimulating factor, interleukin 3, interleukin 1, they give rise to macrophages. If you give them retinoic acid with dibutyryl CAMP, they make muscles. If you give fibroblast growth factors, then followed by fibrillized growth factor 2 and epidermal growth factor, and then later the platelet-derived growth factor as well. You can give astrocytes, oligodendrocytes. So this gives you the, propensity from making iPSCs into any cell type you want. You want to make a fat cell, a neuron, immune cell, or a muscle cell, you can do it all in a dish. So this tells you the power of using this induced pluripotent stem cells. And for example, if you have any mutations in them, you can do gene editing and make healthy cells again of the patient that you have. So this gives you a really huge power that we have now that we can do in the lab. And we don't really need to, go back to an organism or mice to do these studies. So this is a video to show how these induced pluripotent stem cells can make the cardiomyocytes, which can actually beat in a dish. So you can see here, they have been differentiated into cardiomyocytes, and the beating of a heart can be recapitulated just in a dish by reprogramming these iPSCs. And the potential application or the use of these induced pluripotent stem cells for drug discovery or to analyze the treatment of genetic diseases is huge. So you can take the patient-specific fibroblast, you make iPSCs from there. You can see what happens under the microscope when you see the normal progression to the cell type. So for example, if there's a problem in blood cell specification, you can turn these iPSCs to blood cells and see exactly where the defect is happening. Then you can try and block this by different screens of drugs. You can give different kind of NIH-approved drugs to them and see if you can get normal blood cell differentiation from these iPSCs. If not, if you know the mutation by sequencing, you can go in with gene editing technology, repair the mutations in the dish, make the healthy cell, and then transplant them as they're matched to the patient and try to, come about the disease. So this has already been done for sickle cell anemia and has been done in India for sickle cell anemia, where the fibroblasts have been programmed to iPSCs. The gene editing has been done, so the hemoglobin gene is functional again. And then these iPSCs or this hematopoietic stem cells have been transplanted to the donor, and they have replaced the aberrant RBCs, and you can now see functional RBCs in the patient. So actually, this has even reached to the clinic, though this technology right now is really expensive, because as you can imagine, you need to reprogram the cells, do gene editing, then put it back into the patient. It is quite an intensive experiment to do and requires a lot of reagents that are still expensive. But definitely with the time to come, we hope that this technology could be available to a lot of different patients for different, so you can have patient-specific tailored drugs or gene editing technology for your specific rare genetic diseases if you know the rare gene that's involved for the patient. So you have, really patient-specific strategies rather than just a drug that is applied for everyone. So finally, I'd like to sort of talk to you about our institute. So all the topics I've talked to you about today, about developmental biology, stem cells, embryogenesis, induced pluripotent stem cells, embryonic stem cells. We cover all these topics here at the Manipal Institute of Regenerative Medicine in our master's program. It's a 2 years program in stem cell technology and regenerative biology. And so you'll hear from us, who are the faculty, who are part of the Manipal Institute of Regenerative Medicine, but we also get international faculty to give guest lectures. And we have also conferences that are conducted throughout the year at MIRM. The students who are part of the Masters in Stem Cell Technology and Regenerative Biology do a 6 months mini project, followed by another 6-month main project. So they're immersed in the lab for up to 1 year, where they get hands-on exposure on stem cell techniques, and they're exposed to how a lab environment doing research on all the topics I covered are, doing all the cutting edge research in CRISPR-Cas9 or iPSCs or model organisms, all of this, the Masters in Stem Cell Technology and Regenerative Biology, the students get hands-on experience for up to a year. And what are the career opportunities when you do a master's program for MIRM? Well, after obtaining a master's degree, you can register for a PhD program. So MAHE offers a TMA Pi PhD fellowship. So if you do pass the TMA Pi exam, you can be a PhD fellow in either MIRM or any of the MAHE institutes in Manipal or here in Bangalore. But of course, our students go on to go to other institutes such as INSTEM, Indian Institute of Science, JNCS, NCBS, and of course to many international universities abroad. And another factor that we have our pharmaceutical industry. So we do have some sort of possibility of doing internship in different pharmaceutical companies and biotechnology companies. And our students do go on to join these, as well as clinical applications in hospitals. So this gives you a hands-on on how stem cell technology can be used for rare diseases. And the lab of Professor Jyoti is looking at one such rare disease related to blood disorders, and her lab is trying to figure out what exactly goes wrong in these patients to help them, because it's a rare disorder and we don't really have any treatment for it. So hopefully at MIRM, we will be coming with discovering new treatments for diseases which we still don't understand. So with this, I'd like to close my presentation. And of course, I will be happy to take any questions related to our stem cell technology, regenerative biology program, but also my presentation in general. So thank you, Biotechnica, for this opportunity to let me present our program and about a little primer about what we teach in our master's program today. Thank you.
Okay. Thank you so much, Dr. Sveta. So our audience, if you have any questions, feel free to start putting that down in the comment section and we will start picking that up for you. And while you are typing, let me quickly brief you about some factors which you should consider if you are planning for a career in biotech and life sciences. You see, the biotech field demands specialization. And when you have a super specialization at the master's level, your chances of getting a high quality, high paying job is very high. And that's where Dr. Shweta and her team are the architects of tomorrow's biotech scientists who will work on stem cells. Like she told you, Dr. Jyoti, who happens to be the HOD of this MIRM University, MIRM College. So they are working on various rare diseases, and hopefully in the near future, they're going to come up with breakthroughs. And you can get a chance to work with these scientists. Now, how can you do that? Well, you should have a bachelor's in biotech and life sciences, and you should have a zeal. You have to apply for MIRM's master's program, okay? And there will be an entrance test probably or an interview. They will conduct that. And once the pre-screening is done, you will be invited for the program. And this program is affordable. It is high quality. MIRM is in the top 5 colleges in Bangalore as we speak. And the master's program is highly equipped with top-notch labs and scientists like Dr. Shweta. So you should definitely consider a career in stem cell technology and get a chance to work with Dr. Jyoti and Dr. Shweta. Now that we have enough questions in front of us, so Dr. Shweta, I'll start showing them on the screen. I hope you can see the screen. And
I will just exit out of my presentation
yes,
so you can
yes,
see the
yes,
screen. Yes.
yes. All right. So
I can't see your screen actually. I will stop sharing. So
yeah,
maybe
so
you.
you can see the questions on
Yes,
the
I see that.
screen,
Yes, I see
right?
it. Okay.
So there's a first question which says that I have a quick question. I want to make a career in biotechnology. Which major I should select as BS or BSc biotechnology or biochemistry in Canada? Which one is perfect? Can you tell me? So would you like to answer that?
Yes, I think at the BSc level or the bachelor's level, getting a holistic overview of biology is good. So if you specialize already at the bachelor's, maybe it's not such a good thing. Maybe it's
Right.
better to do a biology rather than a biotechnology and then get into specializations as a master's or a PG diploma. That would be
Right.
something that would be better, I think, for career progression. So I don't see a big difference between biology and biotechnology, but I would
Mhm.
definitely say that if you want to already go specifically into biotechnology, I would go ahead and go select biotechnology. Yes.
Right, right. I hope you got that answer all things. Okay. So next question we have is a random question from Miles. Says which is the best college for BSc biotechnology? It's very subjective. It
Very
will be very
subjective.
controversial for
Yeah.
Dr. Shweta to
I
comment
think
that,
I
but
cannot answer this question.
yeah, so we would answer that. We have a list and we are going to come up with that. And also I have already posted on Biotechnica. Please do check it out. That will answer your question. Let's just stick to stem cells, guys, because she is an expert of stem cells. I'm the expert of
Yes.
career part. So
Yes.
yeah. Okay. So next one is I would like to request the team to again post the link so that everybody can fill the form and they can get a free certification course on stem cells. And the ground is already, the stage is already set by Dr. Shweta, but now I would request everybody to fill that form. Okay, in the meanwhile, I'm quickly scrolling and checking out what are the questions. Okay, we have a question from Alam Khan. Hi,
Yes.
sir, ma'am. Currently, I'm 12th class pass out student. I seek to make a career in stem cells research field and
Yes.
decided to take a BSc honors in biotechnology at CSMU University at Navi Mumbai. Okay,
Mhm.
so that's a statement. We wish you all the best, Alam, and
Yes.
yeah, keep shining and
Yes,
keep
I
following
think
biotech.
just a comment here. I think our stem cell MSc students are coming from all different kind of bachelor degrees. I think we are
Right,
not selecting for a specific specialization, really. Our
right,
interview is really to see how interested you are in the stem cell topic and how your knowledge in your field is. We will not be testing you on stem cells already. We will be teaching you about stem cells, right? The interview
right,
will be on your scientific aptitude on what you have learned. So don't worry about the interview process that we don't know enough. It's not a prerequisite for you to come with knowledge about stem cells. We will be, helping you to
right.
get that knowledge here at
Right,
MI.
right. So he continues to mention in the next comment that after
Yes.
my bachelor's, how can I get admission into MIRM on merit basis? So, would you like to answer that?
Yeah, so as you explained, there is a MSc program, so he can apply for the admissions at admissions office, and then there
Mhm.
will be an interview that is taken, and after the clearance of the interview, that admission letter will be issued to you, and then you would just have to register and pay the fees for it. So it's not a kind of complicated; it's quite straightforward. So yeah, it's open right now, so definitely he
Right,
should go ahead and apply, and the interviews are held throughout based on how many applications we have. Usually we have between 40 to 50 students in this particular program, so
right,
definitely we are still looking for MSc students, and the motive of this
mhm,
is to kind of get the word out for recruiting more
right,
students
right.
to our program.
So
Yes.
yeah, I would like to add here, Shweta, that MIRM receives more than 5,000 applications for this program, and it's really difficult to get in. So if in case you want to apply fast and apply early, because later on you guys might miss out, and the team might be busy with interviews and pre-screening, and they might miss out. So please apply as soon as possible. And if you want to know when the notifications will come, it will come on Biotechnica also. So whenever it comes, if you're subscribed to Biotechnica, we'll inform you whenever that comes. Okay, we will shout
And
out.
maybe Biotechnica can also let them know if they want to visit MIRM and see our
Yeah.
cutting edge labs. There is an
Yes.
open day that is going to happen in the month of
Wonderful.
April. So,
Wonderful.
open to come and see the labs. They can see we have world-class cutting-edge labs, and really they will have exposure to whatever they will experience anywhere in the world. Yeah.
Like I said, it's the best, stem cells college in the entire Asia. So you must visit on the open day.
We were the
And
first to offer in Asia a master's
yeah,
in stem cell technology.
first,
We were
yeah,
one of
first,
the,
yes.
yeah, the first. Yes.
Okay. So now the next question is, hello, ma'am. When using Yamanaka factors, are there any ethical concerns when using them in context of diseases and biopharma? Over to you.
Ethical. So actually, so far, the reprogramming using the patient-derived cells is off-target effects, I guess, is what you're talking about. But what we're doing is not really changing the genome of the patient cells. The ethical concerns will happen if you do gene targeting or, changing the genome using CRISPR-Cas9. When you're just transfecting with 4 transcription factors, you're keeping them in the induced form by expressing a protein sort of, by overexpression. So there's not really any reprogramming that is happening, that there is no change in the nucleus. It's the expression of genes that are making them pluripotent. So there's not so much of an ethical concern. It's when you do gene targeting, that is changing the genome, that can have off-target effects, and that is something that is more ethically, which has ethical concerns. Yeah.
All right. I hope that answers your question. Moving ahead, we have another question from AKSJR. Good evening, ma'am. Recently, I hear news that stem cells are made from menstrual blood of our women. I don't think. Okay. Yeah. Over to you.
Yeah. I mean, people have also said the placental blood also has some stem cells and so forth. I think I'm not really aware of the reproducibility of this research, so I wouldn't like to really comment on this. It's not something that is being routinely done in the lab. So, I mean, I maybe there's a one-off or some studies related to this, but I don't know how reproducible this is. So I would, still take this news with a grain of salt. I'm not sure how reliable this is. Yeah.
Right. So I hope that answers the question. Next, we have Rahul Raj, and he's asking, what is the stem cells research future like bioinformatics combined with genomics?
Yeah, so when we do research on stem cells, when we are doing drug discovery or looking at pathway analysis, we definitely use techniques like bioinformatics. I think as researchers, we do wet lab, but we also do dry lab at the same time. So all these go hand in hand. So AI is definitely being used for pathway discoveries in biopharma, and I'm sure it will be starting to be used even in basic research. So there is no such delineation between wet lab and dry lab. You use both for your research. So you need to have exposure of both. For any project, these days you have high data available from all different sources. So students need to be, whether it's a PhD or a master's project, they need to have some kind of acumen in bioinformatics, and they will get that exposure in their master's program as well during the project work. Yeah.
Right. I hope that answers your question, Rahul. Moving forward, now we have next question coming in from Veena Vamshi, and she's asking, can you mention about the treatment of autoimmune diseases like SLE and glomerulonephritis?
Oh, that's a very specific question of autoimmune diseases. So I guess I don't know whether there's any approved treatment for autoimmune diseases like systemic lupus, or the other diseases that they have mentioned that I have seen. But maybe there are some clinical trials related to that. So Veena, I would encourage you to go into the NIH database of clinical trials, and there you would be able to do a search for iPSCs in these diseases, and you should be able to find that information out. As far as I know, specifically in clinical trials, I don't know if iPSCs are used in these currently, but I would encourage Veena to go to NIH website and to look for that information. Yeah.
Yes, and Veena has a follow-up questions asking, she's done a master's in pharmacology. Can she pursue a PG program at MIRM?
Yes, absolutely. We do have a PG diploma in stem cell technology as well. So that would be definitely a 1-year program, which gives you also a mini project of 3 months, where you, of course, are immersed in the semesters just like our masters in stem cell, but you do a mini project of 3 months. And it's a good, bridge for the people who are from clinical or from other backgrounds like pharmacology, and you want wet lab experience and also on basic stem cells. So I would encourage her to apply for that.
Okay. Moving ahead, next question we have is from Vague Life. Says, I've already completed MSc in zoology. Is it too late to get into a specialization?
No, I don't think so. One of my students is a MSc in botany, and I
Mm-hmm.
have hired him as a PhD student. He passed this TMAPI. So MIRM is part of Mahe Manipal, and they have this TMAPI program, which you have to give this Manipal entrance test. And if you clear the test, it doesn't matter whatever your MSc is. We do an interview, and you can definitely enter into MIRM as a PhD student. So no, I don't think so. This precludes you from joining as a PhD student at MIRM. Yeah.
Right. Moving ahead, I think we have an interesting question. Rachna Solanki says that she has been offered admission in Punjab University, Chandigarh, for MSc
Mhm.
Stem Cell and Tissue Engineering, and how it is. Is it a good university? So I think we cannot comment on Punjab University, but we can definitely for sure say that MIRM is one of the best in India and Asia, one of the first in Asia to start this kind of a course, and highly research oriented, and definitely with a very good placement record as well and research record. So if in case you are interested, also consider MIRM. The last date to apply is 5th of April, and the link I will share in the groups right now. Please check that out. Okay.
And talking about the placement, our last batch of students, there's a 85% of placement of our students.
Oh,
So
wow.
we
Wow.
have a
Wow.
very high rate of placement. Yeah. Many of our students have cleared all the national tests. So, our curriculum is quite good to, make them competitive to get any of these national fellowships like ICMR, DBT Net. So yeah,
Right. Right.
students are doing well.
Right. So there's a question from Navodayan says that please keep a session for UG students also. Okay. We'll definitely do
I think
that.
has this not been open for UG students? I made
It
my
was.
presentation accessible to bachelors actually, not for a master student. So I
Yes.
hope it was.
It
Yes.
was. It definitely was, but probably you can rewatch it and you'll catch up.
Yes, it will be recorded, so please rewatch it.
Yes, yes. So Nikita has a question. I'm a biotech postgraduate, currently doing AI/ML internship in Biotechnica. I have background in molecular cloning, protein expression, and purification. How can I upskill myself in stem cell and get a job? What's it?
Yeah, I mean, she is a biotech postgraduate, so I guess that means that she maybe has done a BTech, is what I'm presuming.
Yes.
And now she's doing an internship. So definitely, if she wants to get specific background, I mean, cloning and protein expression, that's good hands-on, but that's not specifically to iPSCs and stem cell technology. So I think, as I mentioned, if she wants to do a shorter program, our MSc program is 2 years, but if she's interested in doing a shorter program, we also have a diploma program as well. So she could look into both of those, and that would give her specialization in stem cell
So
and major.
that's a 1 year program.
That's a 1 year program, yes.
That's a 1
But I
shorter
would
program.
say the curricula in the first semester and the second semester is very much parallel to what we do in both programs. Yes.
Right. So we have a lot of questions right now, but we are running out of time. So we'll just take last question. But
Yes, sure.
it's an interesting question from Dave Puran. Nice name. What are the most promising stem cell based therapies currently in clinical trials?
I think in relation to hematological disorders, it's very promising because you can harvest the stem cells from the blood, edit it, and then put them back. So that's the most promising, and that's where we are seeing most of the success. So when I talked about sickle cell anemia, other disorders like thalassemia, many of them are actually getting treated by using the stem cell technology and gene editing. That's where I feel, the most promise is, because you can just give in transfusion the stem cell back, they will go back to the niche, they will proliferate, and they will give back the normal tissues as well. So that is where I see the most promise, where in the next 5 years, I think already the technology is there in India. It's just a little expensive, but I hope when we make it indigenous and we make it some way that we can do it, in our hospitals in collaboration with the basic scientist, hopefully this technology will be useful for the rest of the population, because we know in India some of these diseases are very prevalent. So that's where I see the most promise.
All right. So with this we come to an end of our session. Just to remind our audience, there's a free stem cell course available today, only for the people who are watching this video, and it is pinned also in the comment section for those who are watching the recording for the next 24 hours. You can avail this and get a free certification course on stem cells, which will create a groundwork for you for a future in stem cells. And if in case you are interested, MIRM Manipal, Mahe Manipal, the excellent, one of the best institutions in our country, is hosting the MSc and PG diploma courses. Now, for bachelor's, if you've done a bachelor's in life sciences, you can go ahead and apply for the master's program there. And if in case you are already a master's, but you want to specialize in stem cell therapy and regenerative medicine, then you can apply for the PG diploma. More details about this will be published on Biotechnica's web pages and newsletters. So stay subscribed, guys. And if in case you have any questions, feel free to write to me at shekhar@biotechnica.org. With this, we come to an end. And now, thank you so much, Dr. Sveta, for joining us this evening, sharing all those insights about stem cells. And I'm personally honored to be hosting you today. And very soon we'll meet in the open day whenever I'm coming out there. So
Definitely.
to our audience, thank you. And Dr. Sveta, it was a pleasure. Thank you so much.
Bye-bye.